Thank you, Michael. So next we have Matthias Schroeter on validating MRI imaging data with serum biomarkers and the whole genome, gene expression data.
Hello, everyone. I'm pleased to be invited to take part in this symposium. Oh my God, what happens here? Red? Okay. Sorry for the long title. I'm coming from the country where we get money just if we write a notice and say, I need $1 million, whatever. However, I want to speak now about how we can validate MRI imaging data with serum biomarkers and gene expression data, such as from the LM Brain Atlas. First, I want to tell something about a neuroskeptical stance in cognitive neuroscience. Second, how we can validate imaging data. Then I want to discuss some experiments on the neuronal marker protein neuron-specific enolase. In the next part, on the glial marker protein S100 beta, and finally I want to conclude. So we have, as all of you know, we have a lot of imaging data. We produce a lot of imaging data. Thousands of studies are published. However, there are still some problems. We have a big knowledge on the macroscopic neural level. We know a lot about diseases, how they change, neurodegenerative disorders, where's atrophy, where changes in white matter appear. However, we do not understand, or we understand only a little, what happens in the same time on the microscopic cellular level, which we can investigate by pathohistology, electrophysiology, molecular biology. So the question arises how we can solve this issue, how we can get access to this mesoscopic level. And I want to suggest some approaches, how we can connect these levels, how we can bridge the macro and microscopic level. And the first one is, of course, animal studies in vivo, where we can investigate all these levels together. We can look for imaging data, we can look for electrophysiological data. After that, we sacrifice the animal. However, in the humans, we have a different situation. We can just have an access to the microscopic cellular level post-mortem. So how we can solve these issues, how we can bridge the gap in humans. We have used meta-analysis, sero and genotyping, and imaging studies and combined them. And I just want to show you some data where we focus on neuron-specific enolase. That's a marker which is found in neurons, particularly neuron-specific enolase, as the name tells. And S100 beta, this is located in astrocytes and in oligodendrocytes. So it's a glial marker for the glue, what has been called the glue in former times of the brain, and has been now associated with many functions, even with brain activation. Okay. This micrograph shows you the localization of NSE in the human brain, and you see here in brown color anti-NSE staining. Neuron-specific enolase is localized in the cytoplasm of neurocytes. It's not secreted, and it's a marker for structural neuronal damage. It has been used as a marker for traumatic brain injury, predictive marker, neurodegenerative diseases, and cardiac arrest. Now I want to jump from cholera to the opposite, to overweight and obesity. There are a lot of studies. It's a very frequent disease, of course. Here is shown gray matter density changes in correlation with the volume of visceral adipose tissue. That's a measure for obesity. Here shown in red, that's in abdominal scans. And you see that there is a tight correlation. So these data suggest that in young subjects you find a decrease in gray matter density associated with overweight and obesity. That's not the case in all subjects. And how can we find an external validation marker for these data, that we can understand what the gray matter density changes mean? And we have used neuron-specific enolase for this to answer this question. Here you see data in the middle row for overweight and obese subjects with a BMI, body mass index, above 25. And you see that gray matter density correlates negatively with neuron-specific enolase, what indicates that This neuronal damage marker indicates atrophy or brain structure changes in the cerebellum in obese, overweight subjects. This is not the case in lean subjects. Going a step further to the SPM anatomy toolbox, these changes have been localized to lobulus 7 and crus 1 and to the entorhinal cortex. And these are very interesting regions because lobule 7 and crus 1 are parts of the cognitive or neocerebellum. We know that lesions in these cerebellar areas lead to executive, spatial, and linguistic deficits, and as shown here in activation likelihood estimate meta-analysis across functional imaging studies, crus 1 is also associated with executive functions and language functions in healthy subjects. Finally, that's another meta-analysis, anatomical likelihood estimate meta-analysis. We know well that hippocampal and entorhinal regions are involved early in Alzheimer's disease and its pre-stages, and one might conclude that overweight, obesity in young age could contribute to later neurodegeneration, such as Alzheimer's disease. So now we went to the Allen Brain Atlas and tried to validate these findings, what I have shown you now. And interestingly, here you see in the heat map expression of NSE, neuron-specific enolase, in the human brain as measured in 2 probes. And you see obviously in the cerebellum hot colors, the red colors, very high expression in the cerebellum across the whole brain, though it's a whole brain approach in the Allen Human Brain Atlas. If we focus to the different cerebellar regions, you see even here crus 1 has a very high expression, lobule 7 as well. So we can validate with the Allen Atlas these findings. What about other imaging modalities? I now just want to speak about resting state MRI, where we investigated connectivity. We used as a measure eigenvector centrality, or eigenvector centrality mapping. That's a data-driven and parameter-free approach. There, voxel receives large values if strongly correlated with many other nodes that are themselves central within the network. That is similar to Google's PageRank algorithm. So these are the data just for overweight and obesity, resting state data. You see again that connectivity in the cerebellum decreases with a higher visceral adipose tissue volume, indicating that connectivity decreases in obesity and overweight in the cerebellum, again in the young human adult and not in all subjects. So one might look also for changes and associations with neuron-specific enolase. You see here data that there is also an association between serum neuron-specific enolase values and connectivity in the cerebellum. Interestingly, only in the young female cerebellum, not in males. Okay. Now I just want to go to another marker, the glial marker protein. This is very relevant for mood disorders because mood disorders have been discussed as glial diseases. We know that frontal and limbic areas are involved in the pathology of mood disorders, that in these areas glial cell density is reduced, and in the other studies have shown that actually oligodendrocytes and astrocytes are involved. So you see a reduction in the density of oligodendrocytes and astrocytes in these regions. On the left, you see a figure showing the reduction in young, depressed subjects in comparison to young controls. That's GFAP immunoreactivity specific for astrocytes. The case is different in older subjects. There you see approximately similar densities of astrocytes. Finally, we know from animal studies that astroglial ablation in the prefrontal cortex induces depressive-like behaviors similar to chronic stress, so a model for depression. And on the other hand, neurons are less obviously affected and changes are very small in comparison to astrocytes. So Rajkowska raised the hypothesis that mood disorders are glial disorders. And how can we prove the glial hypothesis of mood disorders in vivo? And in this case, S100 beta is a very interesting candidate as a serum marker. you can find this S100 beta, as I told you, in astrocytes and oligodendrocytes. That's a double immunofluorescence staining. In red, you see S100 beta. In green, the vessels, vessel staining, and you see that astrocytic end feet cover the vessels. Oops. Okay. So S100 beta is localized intracellularly in the astrocytes and oligodendrocytes. It can be actively secreted by these cells, influences various cellular responses. What is most interesting that it is neuro and gliotrophic, so it's also a plasticity marker. In low concentrations and in high concentrations it might be toxic, but this is not relevant for mood disorders. And it's also influenced by serotonergic and antipsychotic drugs. It's also known to be a susceptibility gene for bipolar disorder, depression, and cognitive dysfunction. So what about the serum levels in mood disorders? We would assume that there are increases in mood disorders due to this glial hypothesis of mood disorders. And as you can see here, these are effect sizes for serum concentration of S100 beta. We have very high values in major depressive disorder and a little bit lower levels in bipolar disorders, but both are very high in comparison to controls. We also know that there was interaction with age. With increasing age, we have higher levels of S100 beta. That fits very well to the histopathological data, what I have shown you before, for depression. And we know that the changes during treatment, the changes are correlated, the changes in S100 beta and the clinical improvement here shown for S100 beta, serological effect size for treatment, and clinical effect size for Hamilton depression scores rating the treatment effects. What about S100 beta in the brain? So it would be very interesting to know S100 beta, is it specific for brain areas? Can we find associations, or is it maybe an unspecific marker? Because you just take one serum marker from one subject, one value. It would be a surprise to find an association actually with brain imaging data. And indeed, you see an association. We have correlated S100 beta serum values with gray matter density and white matter parameters. There was no association with gray matter parameters. However, with white matter parameters here shown, again for females, interestingly only, you see a negative correlation with fractional anisotropy and a positive correlation with radial diffusivity. This is very interesting because radial diffusivity is a specific myelin marker discussed in the literature. There was also significant interaction of sex and S100 beta in these data. Now, returning to the Allen Human Brain Atlas, what about expression in the brain of S100 beta? You see here again the heat map across all brain regions, whole brain regions. And you see a very high expression in the corpus callosum and white matter structures. Here also shown in a bar chart, which fits again very well with the imaging data. So we can again validate with the Allen Human Brain Atlas the imaging data. In these pictures, you see additionally, again, a double immunofluorescence, red S100 beta and green oligodendroglial markers. That's on the left side. You find post-mortem corpus callosum, human corpus callosum. You see in a yellow color the colocalization between both markers, and on the right side a human cell line, again a high colocalization. So we assume, and I think this supports the assumption that S100 beta values are related to oligodendroglial cells. Okay, I just want to conclude now. What has been already done is combining serum biomarkers, MRI imaging and genotyping data with the Allen Human Brain Atlas, and I think that if we extend this field of research, we can bridge the gap between micro and macroscopic cellular and systems neuroscience by including these biomarkers. With these approaches, we can combine studies and can validate animal data in humans in vivo. And on the one hand, we can validate imaging data with the serum biomarkers and the gene expression data, and on the other hand, we can also validate the serum biomarkers, because as I told you, just one measure for one subject, it's a surprise to find an association indeed with brain areas, a regional specific association. Okay, the next step in my mind on the mesoscopic road are extending the serum biomarker spectrum to further cell-specific markers such as MBP or GFAP. These are specific markers for oligodendroglea or astrocytes, incorporating specific serum biomarkers like amyloid tau, TDP-43 in imaging studies with patients. These are especially for neurodegenerative diseases relevant, and adding plasticity markers beside S1 beta BDNF. And finally, the joint analysis of imaging and gene expression data using the Allen Human Brain Atlas and MNI space. I think that's a very ambitious step in the future, as already stated in the first talk. Thank you for your attention, and my co-workers and collaborators.